Welcome back. In today's session, we are going to discuss about cholera. It is a continuation to the topic Epidemiology of Communicable Diseases. The objectives of this session are Introduction to Cholera, Definition of Cholera, the history, problem statement, followed by we will discuss the epidemiology and the epidemiological determinants, which include the agent factor, host factor, and environmental factor, followed by the pathogenesis of cholera, the most important, and it is discussed in detail how the diarrhea comes up in case of cholera, and then the clinical features, vaccination, and lastly the control program of cholera. Let us begin with the introduction. Cholera is caused by Vibrio cholerae. The causative organism is Vibrio cholerae, is a species of a gram-negative facultative anaerobe. You can see it is a gram-negative organism and a facultative anaerobe comma-shaped bacteria. You see Vibrio cholerae being a comma-shaped bacteria given in the picture here. Some strains of Vibrio cholerae are pathogenic to human and they cause a deadly disease called cholera. And this Vibrio cholerae and some strains, we have strains like Vibrio cholerae 01, Vibrio cholerae 0139. So such strains are pathogenic to humans and they are causing deadly disease like cholera. It is derived from the consumption of undercooked or raw marine life species. Are you understanding? The transmission of cholera is by the undercooked food or the raw marine life species or drinking of the contaminated water. If the water is contaminated with Vibrio cholerae and the same water is used for the purpose of drinking, then it can result in the transmission of cholera. It is just the introduction. All of the transmission, the pathogenesis, the various types of Vibrio cholerae will be discussed in detail in the upcoming slides. Is it clear? What did we understand here? The causative organism Vibrio cholerae, transmission through the contaminated water, food, raw marine species, and Vibrio cholerae causes deadly disease called cholera in humans. Definition: cholera is an acute diarrheal disease. It is caused by Vibrio cholerae 01 and 0139. As I said, cholera is caused by both 01 and 0139 serogroups. These serogroups will produce the cholera toxin, which is the primary cause of the cholera. The case range from symptomless to severe infections. You see, in cases of cholera, it can either be a symptomless disease or to a very severe infection. The majority of infections are mild or they are asymptomatic. As I said they are symptomless. Majority of the cases can be symptomless or mild cases, whereas the severe ones are few. And those typical cases of cholera are characterized by the sudden onset of profuse, effortless, watery diarrhea. What do we see here? The diarrhea, what is the characteristic of cholera diarrhea? It will be watery, rice watery, profuse in quantity and effortless diarrhea. If the diarrhea is passed effortlessly, and if it is profuse in quantity, most importantly, it will be followed by vomiting. Is it clear? The diarrhea in the patient is followed by vomiting. Along with it, there is a rapid dehydration. Since there is diarrhea, along with the vomiting, there is a rapid dehydration seen in the patients. There are muscular cramps and suppression of urine. So the loss of the electrolytes will result in the electrolyte imbalance, resulting in the muscular cramps, and there is suppression of the urine as well. Unless there is a rapid replacement of the fluid and electrolytes, the case fatality can be high as 30 to 40%. So it is very important that in cholera patients we need to replace the fluid as quick as possible. It can be the oral rehydration therapy. In severe cases, we will also give intravenous rehydration is done using the ringer lactate solution. So we see that here the rapid replacement of the fluid has to be done, else the case fatality will be high as 30 to 40%. You can see the patient here having a rapid dehydration, suffering from the profuse effortless watery diarrhea followed by vomiting, muscular cramps, dehydration. So these are the typical symptoms and signs which are present in cases of cholera. Moving further to the history of cholera. The bacterium as the cause of the cholera was discovered by Robert Koch in 1884. So we see that Robert Koch is the one who discovered the cholera. The bacterium has a flagellum, a tail-like structure. So Robert Koch discovered this bacteria called Vibrio cholerae to be the cause of the cholera, and he told that this bacteria has a flagellum. You can see here the flagella, and this flagella is a tail-like structure, or the flagellum or the flagella is a tail-like structure. It is situated at one pole of the bacteria, and there are several pili seen throughout its cell surface. You can see the several pili which is seen throughout the cell surfaces here. Okay. And then it undergoes the respiratory and fermentative metabolism with the help of these pili. Is it clear? This bacteria undergoes respiratory and fermentative metabolism. In August, Koch went to Alexandria, Egypt, to investigate the cholera epidemic there. So we see the year in 1884. Robert Koch traveled all to Alexandria and Egypt to investigate the cholera epidemic which was going on there. He found out that the intestinal mucosa of the people who died of cholera always had this bacterium. See, so we see this person, XYZ person, okay? This person is dead because of cholera, and then again we have another person died of cholera, and when the intestines were examined, they always had this bacterium Vibrio cholerae. Yet he could not confirm it as to be the causative agent. He then moved to Kolkata, India, where the epidemic was more severe. Okay, here in Kolkata, the epidemic was more severe. It was from here he isolated the bacterium in the pure culture. So, what did Robert Koch do? He isolated the organism in the pure culture. Subsequently, he confirmed that the bacterium was a new species, and he described this bacterium as a little bent like a comma. The organism is a little bent like a comma. You can see the Vibrio cholera is like a little bent comma shaped bacteria. And then, although the Koch was convinced that the bacterium was the cause of the cholera pathogen, yet he could not entirely procure the critical evidence. So, he failed to produce the required evidences. Hence, the bacterium produced the symptoms in healthy organism was not accepted. Later on, what happens? Then a new Indian physician comes into the role. We will discuss next in the next slide. We see that Robert Koch, though he convinced everybody that bacterium was the cause of the cholera, yet he could not produce the evidence. His experiments which were carried out on the animals did not lead to the appearance of the disease. What was the mistake Koch did? He did not do the human proving. Rather, he went and experimented these things on the animals. Animal proving was done, but the animals turned out to be immune to this pathogen. Hence, it deduced that animals are immune to the human pathogen. So, Robert Koch took this pathogen from the infected person and he inoculated the same in the animal. Turns out the animal is immune. So, now that does not prove that the cholera is the cause of the Vibrio cholerae is the cause of the cholera. Then further what happened? It was only in 1959 in Calcutta that Indian physician Sambhu Nadi he isolated the cholera toxin and showed that it is the cause of the cholera in the healthy subjects. So he showed that the cholera toxin has the capacity to produce the cholera in the healthy subjects. Hence he fully proved the bacterium cholera's relationship. Is it clear? What did Sambunadi do? He introduced the cholera toxin in the healthy individuals, did the human proving, and then he showed the association of this pathogen with the cholera. We move further to the problem statement. The number of cholera cases reported to the World Health Organization are continuing to rise. For 2016, a total of 1,32,121 cases were notified from 38 countries. So we see that how many cases were notified? 1,32,121 cases were reported from 38 countries, including 2,420 deaths. Is it clear? The true burden of the disease is estimated to be 1.3 to 4 million cases and 20,000 to 1,43,000 deaths annually. Okay, the true burden. Forget about the reporting. If we consider the true burden of the disease, we are estimated burden of the disease is 1.3 to 4 million cases. Okay, 1.3 to 4 million cases and 20,000 to 1,43,000 deaths annually. Two serogroups, okay, it is serogroups. Two serogroups of Vibrio cholerae, that is Vibrio cholerae 01 and Vibrio cholerae 0139. Out of which you see the most common serogroup which is responsible for the cholera pandemics was Vibrio cholerae 01. It produces the cholera toxin which is causing the profuse watery diarrhea. We see it is Vibrio cholerae 01. What you have to remember is this, the Vibrio cholerae 01 being the most virulent and it is responsible for the cholera pandemics which produces the profuse watery diarrhea, whereas the Vibrio cholerae 0139 which was first identified in Bangladesh is confined to Southeast Asia. Okay, this Vibrio cholerae serogroup is confined to Southeast Asia and it can cause epidemics, but it is quite less common. Okay, the epidemics is quite less common by Vibrio cholerae 0139, but whereas Vibrio cholerae serogroup 01 is the most virulent. Recent studies indicate that the global warming is creating a favorable environment for the bacteria. Okay, the recent studies have discovered that. Moving further, in India, since the introduction of the cholera, Eltor biotype. So we have another thing, that is Eltor biotype. Eltor biotype is a particular strain of the bacterium Vibrio cholerae, which is the causative agent of cholera. It is also known as Vibrio cholerae biotype Eltor. You can call it either wise. It is a dominant strain in the 7th global cholera pandemic. Okay. The introduction of the cholera Eltor biotype, it was done in 1964. The geographic distribution of cholera in India is considerably changed after the introduction of cholera Eltor biotype. West Bengal, which was previously known as the home of cholera, is no longer called the same. It has lost its reputation as the home of cholera after the introduction of the cholera Eltor biotype. Now this has become a dominant strain, and then it has changed the distribution of the cholera. Many of the states which never had cholera or were almost free from it for a long time now have got infected and they have become endemic foci of Eltor biotype infection. Is it clear? They were previously considered to have no cases of cholera. They were merely having any cases, but now they have become an endemic state of cholera, and especially there is a foci of Eltor biotype infection. During 2017, majority of the cases rather than West Bengal, now they were reported from Delhi, followed by Maharashtra, Gujarat, West Bengal and Rajasthan. So we see that West Bengal has lost its reputation as the home of cholera, and now majority of the cases are reported from Delhi, Maharashtra, Gujarat, West Bengal and Rajasthan. Moving further to the epidemiology. Cholera is both an epidemic and endemic disease. The epidemic and endemic of a disease will depend upon the characteristic of the agent and those of the environment. So, whether it is going to be an epidemic or it is going to be an endemic obviously depends upon cholera the cholera's serogroup, okay, whether it is 01 or 0139 or it is eltor biotype. It depends upon the characteristic of the agent and also depends upon the environment. If it is a hygienic conditions, it is a high income countries where there is a proper sanitation hygiene, then there are low chances of cholera. Whereas if the environment is those of a very poor area, slums, and then if it is containing unhygienic practices, no proper sanitation, then the chances will be high. Global experience has shown that the introduction of cholera into any country cannot be prevented. So, the introduction of cholera can occur in any country. You cannot prevent the introduction of the cholera. It can occur in any country. But the cholera can create a problem only in the areas where sanitation is defective. As we discussed earlier, wherever the sanitation is compromised, the cholera can take place. They have high potential to spread fast and they cause deaths. In area where the cholera is endemic, it's not stable, but it keeps undergoing seasonal fluctuations as well as epidemic outbreaks. It not always remains as an endemic. At times the epidemic outbreaks can occur and also there will be seasonal fluctuations in the cases. Then we are going to discuss about the eltar biotype. The eltar biotype, as I told you, is a particular strain of the Vibrio cholerae, which is the causative agent of cholera. It is the dominant strain in the global cholera pandemic. This, wherever it has spread, it has become endemic as well, with the periodic outbreaks, which is nothing but periodically epidemics also keep appearing. It appears to have a greater endemic tendency. Along with the epidemics, fewer epidemics, it also has a great tendency to cause endemic tendency. The fate of Vibrio cholerae in the inter-epidemic period are, so what happens so basically it is going to cause endemic tendency. What do you mean by endemic tendency? Usual frequency of cases will be there, okay? Usual frequency. Just for an instance, if 100 cases are found every year, next year 88 cases, in and around the same number, okay? Next year 92 cases. So the endemicity will be maintained. At times what happens, an epidemic can occur, 1 lakh cases can come up, so soon it will give up. Epidemic outbreaks can also be seen. So what happens during this endemic and epidemic period? Okay, generally usual frequency endemic disease will be there. Now after some time epidemic outbreaks have occurred. During this interval, what happens is the existence of the long term carriers is seen. Carrier is somebody who does not have the visible signs and symptoms, but they keep shedding the organism in the environment. The existence of the diminished but continuous transmission. There is diminished, yet there is a continuous transmission involving the asymptomatic cases as well. There are few cases which have no visible signs and symptoms. So these asymptomatic cases are also causing the continuous transmission. The persistence of the organism in a free living, perhaps altered form in the environment. It appears in the environment in an altered form. Is it clear? Next, we move further to the epidemiological determinants to the agent factor. The organism that causes cholera is labeled as Vibrio cholerae O group, Vibrio cholerae 01 and 0139. Moving further to the resistance. Vibrio cholerae are killed within 30 minutes by heating at 56 degrees Celsius or within few seconds by boiling. So what do we understand? How is the resistance of Vibrio cholerae? It is killed within 30 minutes. You can kill the Vibrio cholerae within 30 minutes by heating at 56 degree Celsius, and then within few seconds by boiling. The Vibrio cholerae can be killed within few seconds by boiling. Bleaching powder is another disinfectant which kills instant at 6 mg per liter. So the amount of chlorine which is required to disinfect the cholera in the water is, cholera toxin in the water is 6 mg per liter. Moving further to the toxin production. Vibrios multiply in the lumen of the small intestine and they will produce a exotoxin. Since it is affecting the intestines, it is known as an enterotoxin. So what happens? Vibrios after entering the organism, after entering the human, after entering into the human body, it travels and enters into the lumen of the small intestine. Where does it affect? It affects the lumen of especially the small intestine, and there it produces an exotoxin. Since it is affecting the intestine, it is an enterotoxin. The toxin will produce the diarrhea through its effect on the adenylate cyclates cyclic AMP system of mucosal cells of the small intestine. The exotoxin has no effect on other tissues except the intestinal epithelial cells. This you have to keep in the mind, that the toxin will have the effect on the intestines and no effect on other tissues. The previous sentence what I read, I will discuss in detail and then I will come back to this sentence. Then you can understand what actually is happening here. The toxin is producing diarrhea. How is the toxin producing diarrhea in the body? Very important. We need to know what happens in the body. Why is the patient starting up with the diarrhea? Through its effect on adenylate cyclates. So all of this I will discuss in detail. Come back to this sentence. You will have a greatest clarity about it. Hope so. Okay. Let us begin the mechanism of action of cholera toxin. Now I need to imagine that a patient, let us consider here the patient. Now he is very sad because the cholera toxin has entered, via the transmission through fecal-oral route. Let us consider fecal-oral route, that is the contaminated food. He took a contaminated food and water which was containing the Vibrio cholerae. He took fruits, and then this through the fecal contamination has entered into this person. And then what happens? The Vibrio cholerae, this toxin is entering into the small intestine. In the small intestinal epithelium, the microvilli are affected. Intestinal epithelial cells which are having the microvilli, the Vibrio cholerae is entering into the small intestine. Now what happens? This Vibrio cholerae toxin has two subunits. Now we need to imagine that this toxin has entered. The Vibrio cholerae has its toxin, and this toxin has 5 copies of B subunit. 1, 2, 3, 4 and 5 copies of, these are subunit B. 5 copies of B subunit, and it has 2 copies of subunit A. This is A1 and this is A2. There are 2 copies of A subunit and 5 copies of subunit B. So the cholera toxin, you can have a look here. The AB toxin, enterotoxin, has 2 subunits, 5 copies of B subunit which binds with the host cell receptors, and we also have a copy of A subunit. And these subunits will exhibit the toxic activity. Since it is specifically affecting the intestines, we call this toxin as an enterotoxin. Now we see it has a specific action on the lumen of the small intestine. So Each of the 5 B subunits of the cholera toxin, they are going to bind to the glycolipid called ganglioside GM1. Now in the next slide you can see this 5 B subunits. Okay, the 5 B subunits are going to bind to the glycolipid called, there is a glycolipid, and this glycolipid is called as GM1, ganglioside called GM1. And this ganglioside GM1 is found on the surface of intestinal epithelial cells. Is it clear? The toxin went and bind to the GM1. Next, now this binding is going to trigger the endocytosis, where the entire toxin gets internalized into the cell. Now the binding has occurred. The toxin is moving inside, and the endocytosis occurs where the entire toxin has internalized into the cell. You can see it has internalized into the cell. And now next, what happens after the endocytosis occurs, the toxin will travel to the cell's endomembrane system, and eventually it has come to the endoplasmic reticulum. You see, the toxin has traveled through the endomembrane system, reached the endoplasmic reticulum. And after reaching the endoplasmic reticulum, only the A1 peptide, okay, out of the A1 and A2, only the A1 peptide is released from A2, okay, from the A2, the A1 gets released, and the toxin from the lumen of the endoplasmic reticulum, it is transported into the cytoplasm. You can see here, the A1 peptide gets released from A2, and it is traveling from the lumen of the endoplasmic reticulum. This is the lumen of the endoplasmic reticulum. It is entering into the cytoplasm. Okay, it is entering into the cytoplasm. You can see, after entering into the cytoplasm, what is the mission of this A1 peptide? It wants to modify the membrane associated protein called as a G protein. There is a G protein, the A1 peptide. The whole goal of this A1 peptide is it wants to go and modify the membrane associated protein, that is the G protein. And this G protein, generally it binds to an enzyme called adenylate cyclase, and it controls the activity of adenylate cyclase. So we are talking about G protein. What does this G protein do? This G protein, one, it will bind. It binds to what? It binds to adenylate cyclase, and it will control the activity of adenylate cyclase. Next, we see what happens in cholera. Before understanding what happens in cholera, we need to know what is the normal function of this G protein, what is the normal function of this hormone receptors, how is the CAM produced in the body. So we need to know what is cyclic AMP. So generally we know for the hormones and their functions, hormone uptakes and their functions, we have the cyclic AMPs which have the various functions which are carried out in the body. We want the cyclic AMPs. Now we will see what the G protein does. Generally the G protein normally functions in this way. Inactive G protein, there will be inactive G protein, and this inactive G protein normally it will simply reside in the alpha beta gamma complex, okay, the subunit. The G protein is simply residing in the alpha beta gamma subunits. This inactive alpha, now what happens, this inactive alpha subunit, it carries a GDP molecule. When this hormone gets activated, we see now there is some requirement, the hormone is getting activated. Then it leads to the activation of an appropriate receptor. When a hormone activates an appropriate receptor, the hormone comes, it activates its hormone receptors. Then what happens, the appropriate receptor will further activate the G protein, and it allows it to release the GDP and take up the it allows to release the GDP and take up the GDT. Okay, we see here the hormone gets activated, it takes up the GDP and release GTP, just like they take up ATP and ADP and release ATP, an example I'm saying. Similarly, it takes up GDP and releases GTP. Only thing D to T. Okay, D to T. Is it clear? From GD, it gets GT. GDP to GTP. And then we see that the activated alpha subunit, now it has got separated. The alpha subunits will get separated from the beta and gamma subunits, and now the activated alpha subunit will bind and it will activate the adenylyne cyclase. Now it has got activated. It got released from the beta and gamma, and now it has been activated. The GDP is got converted into GTP, and now it is going and binding to the adenylate cyclase. The activated alpha subunit is binded to the adenylate cyclase, and now the adenylate cyclase is activated. It will convert the ATP into CAMP. Now what happens? The activated adenylyne cyclase will convert the ATP into CAMP. ATP gets converted into cyclic AMP. Then the G protein alpha subunits has GTPase activity. Now it has completed its activity, and what happens? It will soon cleave from GTP to again form the GDP. What happens? There is a cleave. Okay, there is a cleavage. What's happening? The GTP again gets converted into GDP, and inactive phosphate molecule will be released. It again gets inactivated. It will go and block this adenylate cyclase. How much ever was required was produced. Okay, so generally what happens? The G protein complex is again reforming. Only a small amount of the cyclic AMP was formed. How much ever was required based upon the requirement of the body, the ATP got converted into cyclic AMP. For this conversion, what happened? Let us quickly revise now. How did the G protein work? First, we were discussing about the toxin. It enters the intestinal epithelium. It enters the lumen, and from the lumen it reaches the ganglioside GM1. Okay, this protein will enter into the cytoplasm via the endoplasmic reticulum. Only the A1 peptide will get released, and after the release of the A1 peptide, this A1 peptide will travel and it will go and bind to the G protein. This G protein will further go and bind to the adenylate cyclase, and binding to the adenylate cyclase, what does it do? The alpha, generally the G protein, we studied about the normal function. Generally what happens? It resides in alpha beta gamma subunit, and now this alpha is required, so it gets detached itself, and then it converts whenever the hormone comes and the hormone receptors are activated. What happens? These GDP are taken up and they are converted into GTP, and after the conversion, what happens? This GTP will go and bind to the adenylate cyclase, and then once the ATP is converted into the cyclic AMP, once the function is over, it again goes back to GDP and the inactive form. But what happens in cholera? So we are supposed to see that what happens in cholera. In case of the cholera toxin, the A1 peptide of the cholera toxin has an enzyme that uses a molecule of NAD plus, that is made up of nicotinamide group, a ribose sugar, and ADP molecule. This adds the ADP ribose molecule to the G protein alpha subunit. So what's happening here? This riboxylase action is happening. The A1 peptide of the cholera toxin, it's being added with ribose and ADP. The ribose sugar and ADP molecule is added to the G protein. G protein's alpha subunit. Okay, and now what happens? This is an ADP ribosylation reaction. Now this is preventing the G protein to cleaving back the GTP into GDP and inorganic phosphate. As I discussed earlier, GDP will be there. It gets converted into GTP, and then it performs its function. ATP will this gets activated. ATP gets converted into the cyclic AMP. Once the function is over, the GTP again cleaves back itself into GDP and inorganic phosphate. But here what's happening? Since the ribose and ADP are blocking the activity of this alpha subunit. No more normal activity is carrying on. There is a block of this cleavage which is not happening. The GTP is not coming back to GDP and inorganic phosphate. Can you see this molecule here? No, it's not happening. As a result, the G protein has been preventing itself from cleaving into the GDP and inorganic phosphate. As a result, the adenylate cyclase remains activated for a prolonged period of time, resulting in the increased levels of the cyclic AMP tremendously. So what happens, since it is binding here, it is not able to cleave, and the cyclic AMP are produced in a tremendous amount. Because of this, the adenylate cyclase is activated for a long period, and the elevated of the levels of the cyclic AMP are stimulating this host enzyme called as a protein kinase. You can see the protein kinase is being activated here. As a result of the elevated levels of the cyclic AMP, they are going and stimulating the enzyme called as a protein kinase A. Now, this activated protein kinase will go and cause the various transport ion channels activation. They will go and activate the various transport ion channels, including the cystic fibrosis transmembrane conductance regulator. So once there is an activation of the cystic fibrosis transmembrane regulator, that is CFTR membranes, that is transport ion channels, alternatively what happens, this will result in the transportation of the chloride ions. Okay, you can see all the chloride ions leaving the cells and entering into the lumen. And then alternatively, even the bloodstream will provide the water, chloride and other ions to enter the intestinal lumen. You can see here in the next slide that including the bloodstream, from the bloodstream the chloride ions and the water leaves the bloodstream and enters into the lumen of the intestine. Why does the water move? In order to maintain the osmolarity. Since the chloride ions are transported, even the water moves out in order to maintain the osmolarity. So the influx of these fluid containing these electrolytes as well will result in the diarrhea. Is it clear? Is it clear how the diarrhea occurred in the body? Do you want a very quick revision? Okay, it is better we repeat multiple times so that there will be a greater clarity and we will not have any confusion. Okay, let us begin. What is the mechanism? Mechanism is the cholera toxin. The cholera toxin entering the intestine. What does it contain? 2 subunits, 5 copies of B, 2 copies of A. Out of which the 5 copies of this cholera B toxin will go and bind to the GM1. What is this GM1? Ganglioside, right? Yeah, it binds to the glycolipid. It is a glycolipid called ganglioside, which is found in the intestinal epithelial cells. Then endocytosis is occurring. The toxin has entered into the cell and it is traveling the endoplasmic reticulum. From the endoplasmic reticulum, from the lumen, only the A1 peptide, okay, the A1 is released, and the A1 peptide is got released, and then it is transported into the cytoplasm. What does it do? It will go to the G protein, which is binding to the enzyme adenylate cyclase. You can see here it is going to the G protein. It goes to the G protein, this toxin, okay, this is important. This toxin is going to the G protein binding to the adenylate cyclase. What does it do? Before going into what it does, you know what it does. When we go directly to the cholera, rather than going into what the G protein normally functions, what happens in cholera, the alpha subunit, it has been replaced by ribose, and we have the NAD molecule, ribose and ADP, and this is blocking the G protein alpha subunit. Now it is not getting converted back. So before understanding that, quickly we need to know how is the normal functioning. In the normal functioning, it is generally residing in the alpha, beta, gamma subunits. Then what happens, whenever the appropriate receptor gets activated, the hormone comes and activates the appropriate receptor, the alpha subunits gets it separates itself from the beta and the gamma subunit, and after the activated alpha subunit gets separated, it will go and bind to the adenylate cyclase, and it will activate the cyclase. ATP gets converted into the cyclic AMP. Then what happens soon after the conversion of the ATP into the cyclic AMP, function is completed. So this alpha subunit will go and block the adenylate cyclase and tell them, okay, our function is done, no more ATPs have to be converted into cyclic AMP. So let us quickly get converted back. Okay, from the GTP, let's quickly get converted back to GDP and inorganic phosphate. But what happens in cases of cholera? In cases of cholera, there is ribosine ADP which is going and binding to this GTP. No longer there is a conversion of GTP into the back into GDP and inorganic phosphate. It is not able to convert. Continuous, there is a conversion of ATP into cyclic AMP. Continuous conversions. There is a rise in the cyclic AMP levels. Increased rise in the cyclic AMP levels is going and activating this protein kinase A. And this protein kinase A, what does it do? As a result of an increase in the cyclic AMP, the enzyme protein kinase A gets activated, and this protein kinase A will open all the conductance regulators, and then all the transmembrane ion channels will be opened. So this opening of the various ion channels, transport ion channels, will lead to the transport of the chloride ions, and not just the chloride ions. In order to maintain the osmolarity, water also goes from the bloodstream into the lumen, resulting in the diarrhea. See, Vibrio cholerae enters the stomach. It shuts down the protein production to conserve the energy and nutrients, and it survives in the acidic medium. So the Vibrio cholerae has the capacity to survive in the acidic environment. In the stomach, it shuts down the protein production, and then once it reaches the intestines, it uses its flagella. In the first slide, when we studied, in the initial slide, we had this flagella. Okay, this flagella will move towards the intestinal walls. It will propel through the mucus layers, and it will attach to the villi on the surface epithelium. There it gets multiplied, and it starts producing the toxins, and these toxins will enter the epithelial cells of the intestines. Now we see the toxin produced will depend upon the strain, whether it is 01, 0139 or LTOR biotype. Okay, then we see that some strains won't cause any symptoms, or they're called mild symptoms. Some other strains, the cholera enterotoxin choleragen, that produces significant symptoms. Okay, and then we are continuing with the pathogenesis. The enterotoxin enters the epithelial cells of the intestine. Now the G protein has permanently been activated, so it keeps activating the adenylate cyclase. It leads to the overproduction of the CAMP, activation of the phosphokinase A, and this will go and open all the transport ion channels, and it will lead to the increased secretion of chloride ions, sodium ions into the lumen, and there is a destruction of osmotic balance between the intestines and the surrounding tissue. Even the water, bicarbonate, potassium will rush into the intestinal lumen. So what all is rushing into the intestinal lumen? Chlorine, sodium, potassium, water, bicarbonate. They are rushing into the intestinal lumen. All this all together is leading to the development of symptoms. So is it clear? So what did I tell you? I will come back to the first slide what we discussed. The sentence I told that we will I'll tell you again. So you see here, the toxin will produce diarrhea through its effect on the adenylate cyclates cyclic AMP system of mucosal cells of the small intestine. Did you understand? What does it do? It will affect the adenylate cyclase and the cyclic AMP system. Okay, there is an overproduction of the cyclic AMP. No stoppage. Okay, and then this is leading to the opening of the channels, and all the ions are entering into the lumen, resulting in the symptoms. Is it very clear? Yeah, we have read it multiple times. You can just quickly go through as I move the slides. You can recall everything on your mind. Yeah. Yes. the pathogenesis as well. So far we have completed so much. Let's move further to the reservoir of infection. The human being is the only known reservoir of cholera infection. It can be a case or a carrier. Case, we see the cases are 75% of the people are infected with Vibrio cholerae. They do not produce any symptoms. As we said earlier, it is symptomless, it is mild. In 75% of the cases, they do not develop any symptoms. Although the bacteria is present in their feces for like 7 to 14 days after the infection, they are shedding back the bacteria into the environment, and it is staying in the cases for at least 7 to 14 days after infection. 20% of the people will develop the acute watery diarrhea with the severe dehydration. What do we understand from the cases? 75% do not develop any symptoms, and 20% will develop severe dehydration. And then we have carriers. Carriers can be temporary, rarely they are chronic. And then we will move further to the infective material. The infective material, the immediate sources of infection is the stool and the vomit of the case and the carriers. We have the cases and the carriers, the stools and the vomits to be the cause of the infection. And then we have the spread of infection. Then we have the infective dose. Cholera is dose related. Infection occurs when the number of vibrios ingested is exceeding the dose that is infective for the individual. So it depends upon the individual. So if the vibrios ingested are exceeding the dose which is required to infect the individual, only then he will develop the infection. So if the cholera vibrio which is ingested are very less, but the person's dose requirement is too much, if the susceptibility is very low, his immunity is very strong, susceptibility is very low to that infection, then he won't be infected. If Vibrio cholerae are quite enough, he's consuming a lot of bacteria, and then that is going to cause him the infection only if the dose required is exceeding. The period of communicability. Cholera is infectious for a period of about 7 to 10 days. Next, we will go on to the carriers of cholera. A cholera carrier is defined as an apparently healthy person who is exceeding the Vibrio cholerae in stools. So, this person will be excreting the Vibrio cholerae 01 in the stools. The carrier is a healthy person. Do you get it? A healthy person, no having the visible signs and symptoms. He will not present with the typical clinical features, whereas he will be shedding the Vibrio cholerae 01 in stools. There are 4 types of carriers. We have incubatory carriers or a preclinical. You see, incubation is preclinical condition. There are no signs, there are no symptoms present in an individual. He is almost a healthy individual who will be shedding the bacteria. So the incubatory carriers are seen of a short duration. They are the potential patients. And then we have the convalescent carriers, the patient who have recovered from an attack of cholera. What is the convalescent period? After recovery, the period after recovery is the convalescent period. So who has recovered from the attack of cholera, but he continues to excrete the cholera vibrio during his convalescent period for 2 to 3 weeks. Okay, when the person, the recovery phase, after the recovery phase, he is still shedding the organism for 2 to 3 weeks. Then we have the contact carriers. This is the infection contracted through association with the source of infection. Contact or healthy carriers are those in whom we see that they are shedding the organism while they are having the infection. This is contracted through an association with the source of infection. Who is the source of infection? We have the cases. And then the duration of the contact carrier is usually less than 10 days. The gallbladder is, if it is not infected, the stool culture is frequently positive for Vibrio cholerae. So we see that in patients, if the gallbladder is not infected, yet the stool culture will be frequently positive for Vibrio cholerae if the gallbladder is completely blocked by some stone or there is some obstructive cause of the biliary system. So the Vibrio cholerae gets trapped there, and then in the whenever it is secreted, there is a positive tests for Vibrio cholerae 01. We move further to the chronic carriers. The longest carrier state was found to be over 10 years. Gallbladder is infected in chronic carriers. So what happens? The gallbladder gets infected in cases of chronic carriers, whereas in the contact carriers, the gallbladder is not infected. Okay, this is the difference what they have made here. Okay, we need not get confused by it. Very simple. We know incubatory carriers for a short duration, and they are the patients, potential patients. Okay, convalescent, they will shed the vibrio cholerae for 2 to 3 weeks after the attack of cholera. Then comes healthy carriers. The infection contracted is through association with the source of infection, and then the duration is less than 10 days. Okay, the contact carriers or the healthy carriers will shed the their period of infectivity will be less than 10 days, and gallbladder is not infected in cases of healthy carriers. Gallbladder is not infected. Okay, cultures will be frequently positive for vibrio cholerae, whereas in chronic carriers the longest carrier state is over 10 years. We see that the carrier stage can last up to 10 years. The gallbladder is infected in cases of chronic carriers. In carriers, the antibody filter against vibrio cholerae 01 rises. Obviously the antibody titre, it is the antibody titer. Okay, the antibody titer against the vibrio cholerae will be raised, and it remains positive as long as the person is harboring the organism in the body. So as long as the person is harboring the vibrio cholerae in the body, the test will remain positive. Even if it is up to 10 years, it can still turn out to be positive as long as the organism is remaining in the body. Moving further to the host factors, age and sex. So the cholera is affecting all the age groups and including both the sexes, that is male and female. In endemic areas, the attacks tends to be highest in the children. And the gastric acidity, we know the vibrio is destroyed in an acidity of pH 5 or lower than that. Okay, if a pH is 5 or lower than that, the vibrio can get destroyed. And comes the population mobility and also the movement of the population. Like during the marriages, there is a movement of the population from the one city to other city, from one state to another state. During the fairs, festivals, these will result in the increased risk of exposure to infection. Then comes the economic status. Where do you think the cholera tends to be higher? It is obviously in the low socioeconomic groups, mainly wherever there is a poor hygiene. Immunity, an attack of cholera is followed by an immunity to reinfection. So whenever there is an attack of cholera, it is followed by immunity to reinfection, but the duration and the degree of immunity is not known. Yes, there is an immunity developed for reinfection, but what is the duration of the immunity which is developed? What is the degree of the immunity is still unknown. Moving further to the mode of transmission, the fecally contaminated water, especially from the contaminated wells, lakes, ponds, streams, and rivers. You can see the transmission can occur from the fecal contaminated wells which are containing the vibrio cholerae in the water, and then we have lakes, ponds, streams, and rivers. Okay, and then we have the contaminated food and drinks. Okay, fecal oral transmission can occur through the contaminated food, through the contaminated drinks, contaminated water, and also these fruits and vegetables which are washed with the contaminated water. If the fruits, if the vegetables, they are washed with the contaminated water, even they can result in the transmission of the infection. And also if the food gets contaminated by the flies, now let's imagine this lady selling the food and these fruits are getting contaminated with the flies, and this is the fecal matter of this infected individual who is having the vibrio cholerae shed in here, and this fly sat on this stool and it carried the vibrio cholerae bacteria in its mouth and again sat on this food, and this fruit was eaten by this healthy person. He took these fruits to his house, and then again everybody will be infected with the vibrio cholerae, whoever are ingesting that fruit. Is it clear? food contaminated with hand, and food contaminated with hand, and food contaminated with flies. Again, hand, how do we explain that? If this lady is suffering with cholera, she has passed stools, or her kid having cholera, she has changed the diapers of that kid and not washed her hands properly, and again she is carrying the vibrio cholerae in her hands, and now the preparation of that chats, let us consider that this guy went and had this fruits or chats or drink from this lady, and now she is going to transmit the vibrio cholerae through her contaminated hands. The flies is explained, hands is explained, food contamination is done. Now comes the direct contact through the contaminated fingers and vomit of the patient, and again, as I explained, through the contaminated fingers and the vomitus of the patient as well, it can get transmitted. Incubation period ranges from few hours up to 5 days, but commonly it takes up to 1 to 2 days. How many hours? A few hours up to 5 days. Okay, it takes 5 days for the appearance of signs and symptoms. If this person is infected, it takes up to 5 days for him to come up with the signs and symptoms, but commonly it takes 1 or 2 days to develop the typical signs and symptoms. Now we move further to the clinical features. The most important aspect, we have the clinical features from a medicine aspect as well. What is the patient present to you? There are 3 stages of cholera: the stage of evacuation, the stage of collapse, and the stage of recovery. In the first slide, we will discuss about the stage of evacuation. The onset is abrupt with a profuse, painless, watery diarrhea. So we know evacuation. So there is an evacuation of the profuse, painless, watery diarrhea, and this evacuation is followed by the vomiting. The patient may pass as many as 40 stools per day. So what do we see here? That the patient can pass up to 40 stools per day. Can you believe it? 40 stools per day. So exhausting it's going to be. Next, we move further to the stage of collapse. The collapse stage, it is critical phase where the body is severely depleted of fluids and electrolytes. Now it is a quite critical stage. Here there is a complete depletion of the fluids, electrolytes, severely depletion has occurred. This results to the various physical signs like the sunken eyes and hollow cheeks. So the sunken eyes and hollow cheeks, the eyes may appear sunken, the cheeks may appear hollow due to the fluid loss. And then we have the scaphoid abdomen, subnormal temperature. The temperature is going to be subnormal. Washerman's hands and feet. They are completely cold. Washerman's hands and feet. Absent pulse. So the weak pulse or absent pulse. Why does it occur? And we also have this unrecordable blood pressure, completely low BP, low blood pressure, weak pulse. The weak pulse, sometimes undetectable pulse, and the low blood pressure, it occurs because of the circulatory collapse. Okay, and then we see there is a loss of skin elasticity. The skin may appear wrinkled. It feels very cold to touch because of the dehydration, and it is known as washerman's hands and feet. And because of the circulatory collapse, there is absent pulse, and there is shallow and quick respiration. The body is struggling to maintain the oxygen levels. As a result, there is a shallow and rapid breathing seen in the patient, since the body is unable to maintain the oxygen level. And decreased or absent urination. The kidneys now are unable to function properly because of the low blood volume. Since there is a lot of dehydration, there is hypovolemia, and it is resulting in the significant decrease or absence of the urine output. Is it clear? And then the patient can also have muscular cramps, particularly in the legs, abdomen. It is because of the electrolyte imbalances. And then we skipped the intense thirst, right? Yeah, the intense thirst. The body's dehydration will lead to an overwhelming feeling of thirst in the patient. Then comes the stage of recovery. The blood pressure will start to begin to rise, and then the temperature will return to normal. Urine secretion will be re-established. The mild cases will recover in 1 or 3 days. The mild cases are going to recover in just 1 or 3 days. Is it clear? BP will be recordable. Temperature coming to normal. Urine secretion will be re-established. Then we move further to the diagnosis. The collection of the stool samples. Stool has to be collected before the person is treated with the antibiotics. We have a rubber catheter. It is introduced into the rectum for at least 4 to 5 centimeters into the rectum, and gently we collect the stool sample. And then we have the rectal swab. The rectal swab, we are going to rub the rectal swab into the rectum gently in a clockwise direction, and this swab will consist of 15 to 20 centimeter long wooden stick with one end wrapped with the absorbent cotton. We see here 15 to 20 centimeter long wooden rod, and then we have wooden stick, and it is wrapped with the cotton which is sterilized by autoclaving. This cotton is sterilized one, and the rectal swabs should be dipped in the holding medium before being introduced into the rectum. And then the vomiters is practically never used because the chances of isolating the vibrio is very less, and there is no advantage. In the vomiters, you cannot isolate the vibrio, so the chances are very less of using vomiters. Whereas the water, 1 to 3 liters of suspected water should be collected in the sterile bottle. We see up to 1 to 3 liters is collected in the sterile bottle, or you can consider it as 9 volumes are collected, and 1 volume is that of a 10% peptone water. Okay, whenever if you consider the well to be infected, well to be contaminated with the vibrio cholerae, so in order to find out the infective organism, the pathogen, what do? We collect the sample, water sample, 9 volumes of water sample, and 1 volume is 10% peptone water. And then we come to the food samples. Food, 1 to 3 gram of the suspected food is collected in a transport media, and again it is sent to the laboratory where the organism is isolated. Then comes the transportation. It is transported in the sterilized McCartney bottles, 30 ml capacity. They are containing the alkaline peptone water. You can see they are containing the alkaline peptone water or the VR medium. If the larger stool specimens will be collected, have to be collected, the VR medium is considered. Next, we have direct microscopic examination with the dark field illumination is available, and it is possible to diagnose up to 80% of the cases within few minutes. Very clearly through the microscopic images, you can see the a small bent comma shaped vibrio cholerae with a flagellum and the various villi. And here you can see this flagella and the comma shaped bacteria. It is very easy to diagnose 80% of the cases within few minutes through the microscopic examination. And then we have culture methods on the arrival at the laboratory, the specimen, whether it is a stool or then we have the like the suspected food, whatever. The stool specimen in the holding fluid is now well shaken, whatever the transport medium, whether the VR medium or from the alkaline peptone water, you will well shake the specimen in the holding fluid, and about 0.5 to 1 ml of the material will be inoculated in the peptone water telluride medium enrichment. So here this medium is peptone water telluride medium enrichment. 0.5 to 1 ml of the material is inoculated, and you can wait and see beautifully there is an easily emulsifiable colonies which are produced. Characterization: vibrio cholerae appears on the bile salt agar as a translucent, moist, raised. You can see they are translucent, they are moist, raised, smooth and easily emulsifiable colonies. They are measuring about 1 mm in diameter. How are they? Smooth, moist, raised, translucent, easily emulsifiable colonies measuring 1 mm in diameter. And then comes the biochemical test, like the production of the colonies in the sucrose and mannose is characteristic of vibrio cholerae. Again you can see here the vibrio cholerae in the TCBS agar, and we also grow it in the sucrose and mannose. Sucrose and mannose is characteristic of vibrio cholerae. Further characterization is done by the slide agglutination test. We are using the anti-01 or 0139 anti-sera. Okay. Lastly, we are going to discuss about the control of the cholera. It is done by the verification of the diagnosis. Like we verified the diagnosis using the culture method, with the help of the rectal swab, and then we have the rubber catheter, culture media, and then direct microscopic examination, biochemical tests, and then we have collection of the food samples and stuffs. Okay, is it clear? And then we are verifying the diagnosis of the cholera. For the specimen diagnosis of the cholera, it is important to identify whether it is Vibrio cholerae 01 in the stools of the patient. Okay, we are identifying the Vibrio cholerae through the stool specimen. And then the early case finding, the early detection of the cases will also permit the detection of the infected household contacts. If we do the early detection of the cases, we can also see the household contacts who are infected, and it helps the epidemiologist in investigating the means of spread for deciding on the specific intervention to be taken. If it is a contaminated well, he can find out how many people in the locality are infected and what is the cause of it, and if it is a well, then he has to do the chlorination using 6 mg per liter. The well needs to be chlorinated, so he can take the steps whatever required to eliminate the source of infection and to combat the epidemic. And then we have notification. Oh, we have some more slides left. Yeah, notification. Since 2005, notification is no longer made mandatory. Health workers at all the levels should be trained to identify and notify the cases immediately to the local health authority. We need to train the health workers that they have to identify the cases and they need to notify the cases immediately to the local health authority. Under the international health regulations, the cholera is notifiable to the World Health Organization within 24 hours of its occurrence. So now the international health regulations have told that cholera has to be notified within 24 hours by the national government. They have made it very clear that it has to be made it notifiable to the World Health Organization within 24 hours. The number of cases and the deaths are also to be reported daily, weekly, till the area is declared free of cholera. So if the cholera epidemic occurs, we need to note down the number of cases and report it, and also the deaths, the mortality also have to be noted and declared, and it has to be reported to the World Health Organization till the area is declared free of cholera. Number of cases and deaths are also to be reported daily, weekly, till the area is declared free of cholera, and the area is declared free of cholera when twice the incubation period has elapsed since the death. Okay, now if the incubation period is 5 to 10 days, we need to see that the 7 days has passed and that twice the incubation period 14 days have passed, and then no deaths have occurred, recovery or isolation of the last case. Since the death, recovery or the isolation of last case, we need to wait for the twice of the incubation period, and then if no cases come up, then we have to consider the area to be free of cholera. Establishment of treatment centers. Mildly dehydrated patients, how do we treat them? With oral rehydration fluid. When we studied about the acute diarrheal diseases, we discussed that the diarrhea, how do the patients whoever come up with the diarrhea, the oral rehydration solution, oral rehydration therapy will be the first line of treatment. If they are mildly dehydrated, 90% of the cholera cases can be treated with the oral rehydration fluid. Severely dehydrated patients who are requiring the intravenous fluids like the anti-diarrheal solution, and then we have the Hartmann solution, ringer lactate. They should be who are requiring these ringer lactate solution have to be transferred to the nearest treatment center or to the nearest hospital. If possible, we need to receive the oral rehydration on the way to the hospital or the treatment center with the convenient distance. The local school or the public building should be taken over and converted into the temporary treatment center. So if the hospital is at a far distance and if the treatment center is not available, so we need to quickly convert, in cases of an epidemics, we need to convert the local school or the public building should be converted into a temporary treatment center. In areas where the peripheral health services are very poor, and the cholera is an endemic disease or it has turned out to be threatening, the mobile terms should be established at the district level. Okay. Now we move further to the rehydration therapy. Mortality rates are brought down to less than 1% by the rehydration therapy. So we see that the death rate is come down to a level of 1%. Can you believe that the death rate is as low as 1% by the effective rehydration therapy? And the rehydration can be oral or it can also be intravenous. Oral rehydration therapy using the ORS solution, and then we have the intravenous using the ringer lactate solution, ringer lactate fluid, okay, IV fluid. And then we have adjuncts to therapy. Antibiotics should be given as soon as the vomiting is stopped. So we are not supposed to give the antiemetics as soon as the vomiting is stops. The antibiotics can be given. It is usually after 3-4 hours of oral rehydration. And what are the common used antibiotics? We have tetracycline, ampicillin, azithromycin, and fluoroquinolones. Fluoroquinolones are used in cases of the bacterial infections. Especially we have ciprofloxacin, levofloxacin, then we have moxifloxacin. All of these are used, and what do? They are bacteriostatic. They prevent the growth of the bacteria. They kill the protein synthesis, the DNA gyrase which is required for the DNA synthesis of the bacteria is cut down. And no other medication should be given to treat the cholera, like the antidiarrheals should not be given, antiemetics are not recommended, antispasmodics not recommended, cardiotonics, corticosteroids, all of these are not required during the cholera. Only an antibiotic called tetracycline, ampicillin, azithromycin, fluoroquinolones can be given, out of which we have ciproflox and norflox. They are the most widely used cholera. They are widely used in cases of vibrio cholerae toxin 01 and 0139. They are used to combat this vibrio cholera disease. Most importantly, out of the fluoroquinolones we have ciproflox and norflox. Moving further to the epidemiological investigations, the general sanitation should be applied. Epidemiological studies must be undertaken. Epidemiologist should maintain contact with all the health and the civic units in his area. Epidemiologist, he has to maintain a good contact with the civic units, to the health and the civic units in his area. Then the sanitation measures. Water control. So we need to provide a properly treated, otherwise a safe water have to be provided to the communities for all the purposes, may it be drinking, washing, cooking. For all the purposes we need to provide a good, properly treated, safe water. Then comes the excreta disposal. Provision of a simple, cheap and effective excreta disposal, that is the provision of the latrines. The latrines have to be provided for the effective excreta disposal. The importance of the hand washing, especially with soap after the defecation, has to be explained to people. We need to encourage them to use the soaps for hand washing, especially after defecation. Then comes the food sanitation. That is how we are going to control the cholera, right? The sale of the food under hygienic conditions. The food has to be sold under hygienic conditions. And health education about eating cooked hot food and proper individual food handling techniques. So the patient should be told that they need to quit eating the stale food, okay? And they have to eat properly cooked hot food, and the food handling techniques, that the proper washing of the fruits and the vegetables before its consumption, and the cooking vessels should be cleaned and dried after each use. Then comes the disinfection. The most effective is a coal tar disinfectant with a Riedel Walker with a coefficient of 10 or more, such as Cresol. So we have the disinfectant, the most effective disinfectant to be the Cresol. It is the disinfectant is a coal tar disinfectant. It has a Riedel Walker along with it. A disinfectant with a Riedel Walker coefficient of less than 5 should not be used. Okay, the coefficient should be 10 or more, such as Cresol is one of the disinfectant example given here. But a disinfectant where the coefficient is less than 5 should not be used. Then we move further to the chemoprophylaxis. What is chemoprophylaxis? The medications which are given or the antibiotics which are administered in the patients before the appearance of signs and symptoms, especially in cases of epidemics. We are administering the antibiotics to prevent the occurrence of disease. It is advised only for household contacts, okay, those in whom the patient is having the capacity to infect the other household members. So if infected individual, their family members can take this chemoprophylaxis, or of a closed community where there is an epidemic, they can take this chemoprophylaxis. And what is the drug of choice here? Tetracycline is a drug of choice for the chemoprophylaxis. It is given over a 3-day period in a twice daily dose of 500 mg for adults. We see how do we prescribe chemoprophylaxis. Tetracycline, it is 500 mg BD, okay, 101 BD for twice daily, and it is given for 3 days. BD for 3 days. Is it clear? Tetracycline 500 mg BD for 3 days. So in adults it is 500 mg in adults, and it is 125 mg in 4 to 13 years. It is 50 mg in age group of 0 to 3 years. A single oral dose of doxycycline 300 mg for adults is recommended. Out of these tetracycline, one of the commonest one used is doxycycline. It is 300 mg for adults, and it is 6 mg. What is the dose? It is 6 mg per kg body weight for less than 15 years. For more than 15 years. Okay. Let's move further to the vaccination. Oral vaccine are of 3 types. We have Dukoral vaccine, Sanchol, Morvax vaccine, and Uvicol vaccine. We see Dukoral, Uvicol, and Sanchol. Let's begin with Dukoral WC-RBS vaccine. It is a monovalent vaccine based upon the formalin. So it is based on formalin, and it is containing the heat-killed whole cells of Vibrio cholerae 01 and recombinant cholera toxin subunits. We know what does vaccine contain? Vaccine contains the pathogen in a killed form. So we see it is containing the heat-killed Vibrio cholerae and the recombinant cholera toxin subunit. The vaccine is also given with a buffer. Since the Vibrio cholerae can get destroyed in the stomach because of its acidity, it cannot survive in a pH less than 5, right? So what happens? The vaccine, in order to prevent its destruction by the acidic medium of the stomach, it is also prescribed with a buffer, and this buffer and vaccine are mixed in a 150 ml of water for the person aged more than 5 years. You can see here a suspension is given, and the effervescent granules for oral suspension is given, and here there is a sodium hydrogen carbonate, a bicarbonate buffer is given, and it is mixed in a 150 ml of water. 75 ml of water for the children aged 2 to 5 years. So we have 150 ml for the children more than 5 years. For less than 5 years we have 75 ml. And then the vaccine has a shelf life of 3 years at 2 to 8 degrees Celsius, and the vaccine can remain stable for 1 month at 37 degrees Celsius. Is it clear? The shelf life of Dukoral vaccine is 3 years at 2 to 8 degrees Celsius. In the ice-lined refrigerators. In the refrigerators it is 3 years, and it can remain stable for 1 month at 37 degrees Celsius. Then the vaccine schedule. Primary immunization is consisting of 2 oral doses, and they are given more than 7 days apart, but it is less than 6 weeks apart for adults and children aged more than 6 years. Okay, 7 days apart. Okay, this is days. Okay, more than 7 days apart. 1, so it should be more than 7 days apart. 8 or 9, it should be more than 7 days apart, but less than 6 weeks apart. for the adults and children aged more than 6 years. For the children aged 2 to 5 years, they should receive 3 doses, okay? 2 doses for children aged more than 6 years and adults, whereas those who are less than 6 years, that is from 2 to 5 years, they receive 7 doses. Again, that is 7 days apart and less than 6 weeks apart. Intake of the food and the drink should be avoided for 1 hour before and after the vaccination. Also, this is important that the intake of food and drink should be avoided for 1 hour before and after vaccination. Then we are going to discuss about Sanchol vaccination. So the Sanchol vaccine, these doses do not contain the bacteria toxin B unit. So this bacteria toxin B unit has the has that thing that it gets destroyed by the acidic medium. Since this Sanchol vaccine does not contain the bacteria toxin B unit, therefore it does not require any buffer. It is administered orally in 2 liquid doses 14 days apart for individuals aged more than 1 year. So again, this is administered 14 days apart. Sanchol is 14 days apart, whereas Ducrol is it is 7 days apart, right? We see Ducrol is 7 days apart. We saw it is 7 days apart, more than 7 days apart. Here it is more than 14 days apart for the individuals aged more than 1 year. A booster dose is recommended after 2 years. Then we have Uvchol. This vaccine is pre-qualified in December 2015 and it has the same characteristic of Sanchol. Okay, it is recently pre-qualified, so we are not very aware of Sanchol, Uvchol. So these vaccine is pre-qualified in December 2015. What are the characteristic of Sanchol? It is Uvchol. It is similar to that of Sanchol. What is it? It contains the cholera toxin B subunit, bacterial toxin B subunit. Since it does not contain the bacterial toxin B subunit, it does not require the buffer. So this cholera toxin B subunit can get destroyed in acidic medium, so we give the buffer. Since it is not containing, no need of the buffer. Then comes the health education. The effectiveness and simplicity of the oral rehydration therapy has to be educated to the people. We need to tell them that the ORS is the most useful in cases of the diarrhea, in cases of the cholera. The patient has to be rehydrated as much as possible, and then it is 75 ml per kg body weight. And then we see that into the age of the patient, and then we see that we take the weight more than the age. We consider the weight of the patient. Okay, weight 75 ml per kg into weight of the patient is the formula which is used for the administration of the oral rehydration solution. And we see that this is the most effective in cases of cholera and diarrhea. 90% of the cases can be dealt with the oral rehydration solution itself. And the benefits of the early reporting for the prompt treatment: as soon as the reporting is done, no need if we treat the patient in the earlier stages of evacuation, the patient will not go into collapse, so it is very easy. The mortality rate can be reduced. The food hygiene practices, as we discussed earlier, that the proper handling of the food, they are not supposed to eat the stale food. The sale of the food should be done in the hygienic manner. So all of these have to be explained to the communities. The awareness have to be spread, and the hand washing after defecation and before eating. Even in the school camps, we need to educate the children about the hand washing after defecation. Even the mothers have to be educated that after changing the pampers or the diapers of the kid who has passed the stools, the mothers have to do the hand washing with the soap. And the benefits of cooked hot food. Okay, the cooked hot food, since the bacteria dies or it can be destroyed by boiling for few minutes at 56 degrees Celsius, the bacteria is destroyed. So the benefits of the cooked food, hot food, proper cooked food will destroy most of the bacteria. And then the safe water and the use of the safe water for all purposes such as drinking, cooking, bathing. So for all purposes, the safe water or the well treated chlorinated water has to be supplied. So this is all about the sanitation measures and the health education. Now we are moving further to the diarrheal disease and control program. The diarrheal disease control program was started during the year 1989 and 1991. This strategy of the National Cholera Control Program has undergone the changes. So the disease diarrhea control program during the year of 89 and 91, the National Cholera Control Program has undergone the changes, and now the National Cholera Control Program is changed into Diarrhea Disease Control Program. From the National Cholera Control, it has been changed into Diarrhea Disease Control Program. Oral rehydration therapy program has been started in 1986 and 87 in a phased manner. What is the objectives of this oral rehydration therapy program? That objective is to prevent the diarrhea associated death in children due to dehydration. We want to prevent the deaths which are occurring due to diarrhea, especially leading to dehydration and death in the children. ORS is promoted as a first line of treatment. This is very important. ORS is promoted to be the first line of treatment in cases of the diarrheal disease control program. They have discussed that it is the first line of treatment. So we need to educate the people to make use of the oral rehydration solution in cases of diarrhea and cholera. Thanks for watching. With this, we come to an end of the session. If you like the video, hit the like button and subscribe.